Yeah, Bismillahirrahmanirrahim. Today's topic is the normal human microbiome. Sorry, department. So, topic is normal human microbiota or microbiomes. The term normal flora denotes the population of the microorganisms that inhabit the skin and mucous membrane. A healthy, normal person. The human microbiome consists of 10 to 100 distinct symbiotic microbial cells harbored by each person, primarily bacteria and the gut. These are the microbiota composition in different region of the body: respiratory, skin, vagina, gut, and oral, what is normal flora. Normal flora are the microbiome, mostly bacteria that continuously inhabited the human body. Under normal conditions in a healthy human, they are harmless and maybe even beneficial. Also called commensal, it is organism that dines together. The beneficial role of flora present in the human body are provide defense against microbial pathogens, helps in the digestion of food, contributes to development of immune system, protection against the potential harmful microbiome, stimulate the immune system, flora of intestine stays stable, beneficial to both human and bacteria. The disadvantage, they can cause disease in the following: when individuals become immunocompromised or debilitated, or when they change their usual anatomical positions. The disadvantage can act as an opportunistic pathogen, ensure nutrients and drug resistant with the pathogens, may be a source of infection to other individuals. Advantages are prevent colonization by pathogen, synthesis and secreted vitamin K and B, stimulate antibody-mediated immune response, inhibits and kill pathogens with production of enzymes like peroxidase enzymes, bacteriocins, no specific fatty acids. Benefits of the normal flora: synthesize and excrete vitamins like K and B12, prevent colonization by the pathogens, may antagonize other bacteria, and they stimulate the development of certain tissues like intestines and lymphatic tissues, stimulates the production of the cross-reactive antibodies. Low-level antibodies produced against the components of the normal flora are known to cross-react with a certain related pathogens, and they will prevent infection or invasions. The normal microbiota are the microbes typically found in healthy people. These microbes are present in various places. The body may be pathogenic, causing disease, and they are not in the process of doing so. Many organisms in the normal microbiota are considered fortunate pathogens. These are the microbes that normally do not cause disease in the healthy persons, but can cause disease when the host is compromised, lack a fully functioning immune system uh, due to treatment with the immunosuppressive drugs or genetic defect in some components of the immune system, or infections with agents and target cells of the immune system. Partial pathogen can also cause disease in immunocompromised host when introduced to an unprotected site. For example, epidermidis is normal background on the surface of the skin. However, when there is a break in the skin, the bacteria may enter to the deeper tissue and even blood, and when they cause infections. E. coli is a normal, another example. E. coli is a normal habitant of GIT when enter the urinary system, causing UTI. A classic example is genital infection of women by Neisseria gonorrhoeae or chlamydia. A typhoid carrier also. In the latent infection, the infection agent is present, but not actually growing or growing extremely slowly. Under the certain condition, the action may reactivate and the organism begins to grow. This usually results in disease symptoms, and the infections can now be spread to others. The classic example is tuberculosis. Probiotics and prebiotics. Probiotics are live microbial supplements, and it benefits the host by improving intestinal microbalance visibly by affecting the composition of the intestine. Probiotics are not drugs. The live organisms which, when administered in adequate amount, confer health benefits on the host. In general, probiotics are composed of bacterial species in the genera Lactobacilli and the Bifidobacteriums. Prebiotics are non-digestible dietary supplements that modify the balance of the intestinal microbiome, stimulating the growth and activity of the beneficial organisms. The term dysbiosis is often used to describe the state where there is interruption or shift in the balance of organisms in the microbiota. Another example of dysbiosis is antibiotic-associated diarrhea. Antibiotics kill many but not all of the bacteria in the GIT, when can allow other bacteria to grow and cause disease. The most important common members of the GIT biomotor associated with antibiotic-associated diarrhea is cholesterol deficit, which is also known as C deficit, and is caused by the antibiotics like the clarithromycin and so on. This causes the pseudomembranous colitis, which is accompanied by severe diarrhea mucus and accompanied by mucus and diarrhea and blood also. Are all over us. There are millions of microbes per square inch on our body, thousands of species. Stare site of the body, places in the body where there is no little residence microbe. In some cases, a few microbes may be transiently present, but they are not really permanently colonized and transit. Microbes are declared rapidly by the body in defenses. The residence flora, fixed type of microbe, regularly found in the given area at a given age. If disrupted, it promptly re-establishes itself. The transit flora consists of non-pathogenic microorganisms and inhibit skin or mucous membrane for hours, days, or weeks. In the underlying sites are places where I never find microbe healthy, but like stomach, lower respiratory tract, kidney, digestive gland, pancreas, gallbladder, CNS systems, and meninges, skeletal muscles, cardiovascular system, poetic, reticular systems, flora, peritoneum, urinary system, bladder, urethra, and genital system. These are the sites that harbors the normal flora. The figure shows. Commonly identified bacteria of the normal microbiome body side. Skin, where few gram-negative organisms fertilize the skin because they do not thrive well in the dry, high salt environment. Present microbes include Staphylococcus family and Propionibacterium, Micrococcus, Corynebacterium, and Acinetobacter. Transient microbes are frequently washed off Staphylococcus clostridium, Perkinges, and Bacillus. In the eye, small number of bacteria due to protective nature of lysosome in tears. The Staphylococcus species and Neisseria species, Haemophilus species, few members of the Rhizopharia microbiota. In the air, there are Staphylococcus species, coagulase negative, Streptococcus pneumoniae, Pseudomonas aeruginosa, and Enterobacterium like E. coli, Klebsiella pneumoniae. In the upper respiratory tract, nasopharynx and oropharynx, there are Streptococcus, Neisseria, Haemophilus influenza, Mycoplasma, Corynebacterium, Enterobacterium. The lower respiratory tract is protected by the mucociliary blanket and only transit inhaled organisms from the upper respiratory tract received here. In the mouth, a lot of anaerobes are also found in the gums, like Streptococcus species, Virulent Streptococci, Neisseria species, Diphtheroid Corynebacteria, Staphylococcus aureus, Epidermidis, Lactobacilli, Mycoplasma, and Spirochetes. And the Streptococcus mutant is found in large number dental plague as precursor of the carriers. The entered bacteria produce large amount of acids, they demineralize the enamels and initiate the caries. In the gums, we can find bacteroids, Bacterium, Clostridium, and Streptococcus. In the GIT, the oesophagus, very few permanent organizers, most transit organisms from the mouth and upper respiratory tract. Stomach, in the stomach usually sterile due to the presence of acid and enzymes, especially when fasting. Occasionally there will be 103 colony forming environment of acid resistant microbes, Streptococcus species, Lactobacilli, and Helicobacter pylori. In the small intestines, the concentration of bacteria increases with the distance from stomach and more are anaerobes like Enterococcus species, Streptococcus, Poromonas, Previtarola species. The small intestines contain a number of Streptococci, Lactobacilli, and normal flora of the intestinal tract plays a significant role in extraintestinal disease, example, E. coli, Enterococcus causing urinary tract infection, bacteroides causing peritonitis associated with perforation of the intestinal wall. In the large intestinal colon, bacteria mass species is 25% by weight. The organisms are probiotics, bifidobacterium, eubacterium, bacteroid, Fusobacterium, Clostridium, Lactobacillus, and Enterobacter family. And the male genital tract generally is sterile, except for the last 1 to 3 centimeter of anterior urethra. Streptococcus species, purple is negative. Streptococcus species, Lactobacillus species. In the female urogenital tract, as the urinary tract is usually sterile, and the vaginal tract varies with the hormone level. The cervix and the uterus are usually considered sterile. Pre-menarche and the post-menopausal sparse colonization usually derived from the skin and colon. Streptococcus species, Streptococcus species, Enterococcus, and Enterobacteria. During the year when estrogen is produced, the deposition of glycogen in the vaginal epithelial cells supports the growth of lactobacillus species, which produce lactic acid, leading to an acidic pH and growth of specific microbe. They are lactobacillus species, Streptococcus, agalactiaceae, guardian of vaginal, mycoplasma, uric plasma, Clostridium species, Enterococcus species. Thank you.